Hello, my students. This is the part B of lecture of part of chapter 11. And this part of the lecture will be focused on structure and replication of DNA. For this particular part of the lecture, there are 3 pages for the lecture note sheet. You may notice that in this particular lecture note sheet, there are going to be different sets of questions that are going to be given to you. Let me try to clarify some things for you. So when you take a look at the first page of this lecture note sheet, you will notice that here is a diagram of the DNA structure, which I need you to complete as you are watching the lecture. But this portion of the lecture, it's also best for you to complete this diagram when you are watching this particular video that talks about the structure of the DNA. And these are also some of the additional questions that is used for guiding in your understanding. So you could choose to answer some of these questions as I go through the lecture, or you could answer it after you watch the video. On page 2 of this lecture note sheet, you will find a continuation of some of these questions, which I have broken it down so that you can follow the information with a little bit easier. And then there is a second video that I want you to watch called the Meselson-Stahl experiment, which is considered as one of the most beautiful biological experiments that has been done so far. And what I have done in here is that I want to trigger your memory from some of the concepts in biochemistry that you have learned back in the fall semester. So you see some questions related to protons and neutrons that will be popping up, as well as some of the basic scientific concepts that you should be familiar with in terms of scientific design for a controlled experiment. And here is some actual experimental data that you would see. If you have any trouble visualizing this particular image, take a look at the image that is inside the recording itself when the lecture is going on, or you can also simply Google this underneath Meselson-Stahl, and you can see a enlarged color image of it. And then on page 3, you will see that here is a relatively simplified overview of how the DNA replication process takes place. Please remember to take your own notes, write in your own words. Please do not download similar types of images and then copy it onto your lecture note sheet. That will not suffice if you want to get a high grade for it. You will likely be getting an incomplete from me if you did that, because I really want you to show me that you understand the material. Now, you will see that on page 3, there are only 3 questions associated with this particular video clip. But the reason why you only see 3 is because all of this blank space that I have left here is for you to complete question 1 that is on this clip. That is to draw the process of a DNA replication in greater detail, but using your own drawing and labeling. And you also need to provide a brief caption for the function of each one of these enzymes, which will be clearly described inside the video clip as well as during the lecture as well. All right, so let's begin. So what is the structure of DNA? Up in this point in time, you have already completed your exit challenge from lecture A, and you have already learned about the vast number of teams of scientists who has contributed to the knowledge that we now know of as the structural model of the double helix itself. For many of you, you may be quite surprised when you look at that particular article that I had assigned you to read, is that they were a significant number of scientist teams that were involved in making different levels and scales of discovery, which ultimately pretty much resembles like a pieces of a puzzle, that when you put them together, that's how Watson and Crick came up with this model. So it's not as though there is a team of just 2 scientists that figured out everything. That really was not true. And I hope that you had fun uncovering names of other scientists who were not always fully recognized in our reading textbooks, where you get to read about who made the other discoveries that were also involved in uncovering the structure of the DNA. So what exactly is the structure of the DNA? Well, DNA is made up of 4 different kinds of nucleotides, which are composed of the sugar, phosphate, and a nitrogen base. Depending upon the type of nitrogen base that is making up part of this nucleotide, we will call it the adenine nucleotide, thymine nucleotide, cytosine nucleotide, or guanine nucleotide. The structure of the DNA itself is a double helix that is made up of 2 nucleotide strands. And within the double helix itself, it is made up of large numbers of hydrogen bonds that holds the complementary base pairs A bond to T, C bonding to G that forms the rungs of this double helix, or as some people would describe, is the rungs of a ladder. And the two sides that forms the ladder, where, you know, if it's a real ladder that you and I can climb on, where we put our hands on, where we hold on to, those are actually formed together with the sugar and phosphate groups. And they are held together using what is known as the phosphodiester bonds. So these are different kinds of chemical bonds that are used to hold all of these molecules together. In your previous reading, you learned, and that's also from watching the NOVA documentary, Rosalind Franklin actually played a significant role in the understanding of the structure of DNA. During that period of time in the race towards figuring out the model of DNA, there were different kinds of proposals. Now, one of the proposals that was even earlier than Watson and Crick was actually devised by Linus Pauling. Linus Pauling is a Nobel laureate himself. This was the model that he proposed, and as you could see, what he proposed is quite different from what we now know as double helix. Initially, he proposed something called a triple DNA helix model, in which you can see that the sugar group, the 5-carbon sugar that you see here, indicated by the blue color portions of this diagram, and the phosphates were held together on the inside of the double helix, as opposed to being on the outsides that form the sides of the ladder that we know of today. And he placed all of the bases on the outside of the ladder. The problem with this is that the chemical bondings themselves would not be stable enough to hold the DNA molecule together. It was quite unfortunate that it didn't work out for him. Otherwise, this would have been another huge discovery in his scientific career. But the work of Watson and Crick, in the large part, owes to the photograph, the famous photograph that Rosalind Franklin has taken, called photograph number 51. This is a bDNA molecule, and what's really special is that if you look at this particular image, where you see the reason why Watson and Crick was able to fairly quickly figure out that the structure of the DNA has to be a double helix and not triple helix, like what Linus Pauling had proposed, is because of this particular image. You can see from this image that the DNA itself is made up of different numbers of major groups and minor groups that twist and turn. And the distance in which these turns would occur could be measured using some of the spots that you can see that is indicated in her photograph. Now, I'm not going to divulge any more on this since you already watched a documentary on this. But let's carry on and take a closer look at this. So at the time when Watson and Crick published their work and created this model, they were both very, very young. And in addition to it, much of the work that they have uncovered actually led to the subsequent major explosion of the biological fields in biochemistry and genetics, which allowed scientists to go on and figure out how inheritance of body traits occur, and how can changes to these genetic molecules, which we now call mutations, can lead to various changes in phenotypes, as well as the onset of diseases as well. So here is a simplified diagram of the DNA model itself. Now, in diagram A, what you see, it's like taking a double helix, which is twist and turn like a spiraling ladder, as is most commonly seen, like this image right here in the middle. Say if you can unwind it somehow and you can flatten it, what you will find is that on both sides of this ladder, it is held on together by sugar and phosphate. Now, the sugar is this 5-carbon sugar that you see here, and the yellow dots represented by the phosphate. They alternate consistently, phosphate, sugar, phosphate, sugar, phosphate, sugar. And the same is true on the other side, that you see this alternation. But right on the inside of the rungs that makes up the double helix or the ladder, it's actually held together by different numbers of nitrogen bases. And what was uncovered was that adenine always binds with thymine, and a cytosine always binds with a guanine. So if you recall from the end of the previous lecture, what you have is purine binding with pyrimidines because of the hydrogen base pairs that they have that actually works together very well. I want you to take a moment and look at this particular diagram. One of the things that I have shared with you in the past is when you look at these schematic diagrams in a textbook or anywhere, it is always designed for a particular purpose to illustrate a particular point that the author wants to get across. But sometimes it could be a bit misleading. So if you look at the structures that is written on here, can you see something that doesn't quite make sense? All right. Now, you know about the structure of DNA. How are they arranged? Now, one thing I can help you with is this. Take a look at A. This is adenine, right? Adenine, well, we're keeping it very simple. Supposedly has a curvature structure like this rounded back reverse shaped C that you see here, right? From adenine. And then you have thymine that will complementary base pair this. And then you have cytosine that is here that has a V shape, but then the G, guanine, has a more sharpened shape like that. In reality, the way this is drawn is just to make it easier for your eyes to see. But what really is going on is that if you take a look at this, and if I were to give you all of these pieces of A, C, G, and T, think about the A. You see how the A is letter A written here? If I want you to fit this piece up next to this letter T right down here, wouldn't you have to turn this letter A molecule? Imagine if this was on a piece of paper. Wouldn't you have to flip it over so that it is upside down in order for you to have this curvature fit right down here in here? And then for the for the letter for the C, the guanine that you have here, right? This letter G would have had to be turned upside down in order for it to fit. In reality, because DNA strands on both sides are anti-parallel to each other, these letters, if they were to have it really represented very clearly, it should have been drawn like this upside down and labeled for you. So I just want you to understand, when you look at the textbook and you see the letters all written upright, that's just for ease of reading, but it really does, it's not technically correct. In reality, the molecules actually will have to be bent backwards for it to fit. Now, this is a really, really nice video produced by MIT regarding the structure of DNA. This is the one that is corresponding to the list of questions I have given you on page 1, floating into page 2. Please watch the video itself and answer the questions related to the structures of DNA itself. Now, I want to go back to this a little bit for a moment. By looking at the structure of DNA like this right now, and I can even do it from this one. Let me go back to here, right? The two sides of the ladder itself, where it's constantly going to be the same, sugar, phosphate, sugar, phosphate, sugar, phosphate. Do you expect the genetic information to be encoded on the two sides of the double helix, or would you expect it to be encoded in the center right here? Now, if you're thinking that no way, because the two sides of the ladder are always going to be the same between even different species of organisms, then you are right. The genetic information is not encoded on the two sides of the ladder, but it's instead encoded in the rungs of these ladders. Now, some of you may be thinking, but how could that be? A is always paired with T and C is always pairing with G. How different can it be? What different is, how it makes it to be different is the combinations of letters, of bases, I should say, that forms a particular gene. For example, you and I can speak the English language, and we all use 26 alphabets to form different words. Depending upon which of those combinations of letters we use and the length of the letters themselves, they will convey a different word, which has a different meaning. And then when it's used and formed into a sentence, into a particular sequence and order, that will convey a particular meaning and comprehension on the other person who's receiving your information. So that is how actually works. So for any given gene that is found on a chromosome, they will have different combinations of nitrogen base pairs that are essentially determining the phenotype that will be expressed in the next generation. So if you have a particular patterns of ACTGs that are formed into a particular segment that codes for one blood type, if you change the order or sequence of it, then you will have a different blood type or a different Rh factor. That's the point. We're going to come back to this and take a look at how that affects mutations a little bit more later on. So now, with the structure of DNA actually being uncovered by Watson and Crick immediately already hypothesized that based on the structure of the DNA, since it's made up of two strands, that when you copy it, most likely it would be semi-conservative. What it means is that they are envisioning that the DNA molecule somehow would open itself up from the two sides, unzip itself like a zipper, and so that you can make copies of either side of it. However, that was just a hypothesis that they did not have any type of solid data to back themselves on, but it was their hunch. And as a matter of fact, during that period of time, when many different competing teams of scientists were trying to figure this out, three theories were, or postulates, if you will, was most popular at the time. Many scientists believe that it could either be one of these three approaches. The DNA molecule probably copy itself using a semi-conservative model. What that means is that you see this twisted double helix right here, and it's made up of two strands that are intertwined with each other. They envision that they can somehow unwind and open up such that you can make a copy of each side by using the original strand, which is indicated in the dark red solid color right here, as a template, like as a mold, where you can use the new nucleotides to base pair with it so that you form the new strand. And by the time you're done unzipping the entire structure itself, you would then have two copies of the original DNA that will be identical to the first one. All right, like this. This is a simple schematic diagram. However, there were other competing teams of scientists who believe otherwise. They believe it most likely would use a conservative model instead. Conservative model means that the DNA will only make a copy of itself originally, and the new ones will be completely so essentially you don't use the old template. You don't use the DNA, the original strand as a template, but instead it would just somehow make a full copy made up of all new nucleotides on both strands, but somehow they would be identical to each other. That was the other thought. The third idea that was proposed at the time was that well, probably you would have portions of the DNA that would be kept exactly the same, and then other portions that are completely newly synthesized by whole new chains of nucleotides. Now, let me try to clarify one or two things in your mind. You may be wondering, well, where are these nucleotides coming from? Remember, we eat food. Food provides the raw materials for us to build our body parts. So there are free-floating nucleotides that are kept inside our cells that can be used to make new molecules. And so, but the question here is, well, how do you make the copy of this? And remember, these proposals are made without having anyone actually seeing any one of these processes happening. So these are proposals, these are thoughts that they have at the time where they're still trying to figure out a method in which you can prove that your idea is right. But this holds significance to it because this is where inheritance happens, which is why this portion of the experiment, if anyone figures out, it's going to be a big deal. And this is where Meselson and Stahl's experiment comes in. Meselson and Stahl are two different scientists who actually had very different research interests. But the one thing that got them interested with each other is the interest that they both want to figure this out. How does DNA replicate? Because it's a fundamental biology question that hinges onto inheritance and how traits are being passed on onto the next generation. So let me help you out with a little bit of this. Now, this is, again, a simple schematic diagram. I want to give you lay a little bit background foundation for you, but then I also have prepared for you a video clip that I thought has done a pretty good job in explaining how this all works so that you can understand the data that you're about to analyze. So what Meselson and Stahl propose is, let's come up with an experiment where we can prove and compare all three ideas at the same time. So what would it be? How can they do it? Because you can't really see DNA and manipulate it directly. It's not as though you can put it under a microscope and watch what happens. So what happens then is that they took bacterial DNA. In this case, is Escherichia coli, which is essentially E. coli that you and I have probably heard of this type of bacteria very often. They take this bacteria, which happens to duplicate every 20 minutes, meaning it would reproduce new cells every 20 minutes. All right, that's a really good idea. And you can put it and keep it and grow it inside an Erlenmeyer flask, and you can grow significant numbers of it as long as you provide it with sufficient amount of food and you give it the medium that it wants. So then what they did was they take the E. coli, they found an isotope, all right, that can latch onto the DNA itself, all right? The isotope in this particular case is N15. Now, DNA molecules contain nitrogen, which is why they chose the isotope of N15, and then the other one, which is a lighter one, called the N14. What they did was they take the the E. coli, grow it inside two separate flasks. All right, one of them, they would expose it to N15. The other one, they would have it expose it with the N14, let it grow. So how does this work? Remember, what we're trying to figure out is with the new DNA double helixes that would be formed, how would they incorporate the N15 compared to the N14? Now, the thing is that each one of these two isotopes carry a different density. So one is heavier than the other. N15 certainly is heavier than N14. And by comparing the difference between the two of them, you can use it to figure out whether, based on the density, the overall DNA that comes out, that are newly formed, which one of these three competing ideas makes more sense. So what they then did is that they grew some of the E. coli using giving them this, another jar of E. coli that was grown in here. And then what they did was they then combined them to see what the organism would do, and then let it go through several different generations. Now, generation sounds like a big word in this case, but remember, these bacteria reproduce every 20 minutes, which is fabulous for this type of genetic study, because then you can have many generations that you can do this with. And because it doesn't take much space, you can keep a lot of these samples in your laboratory and run it over and over again. All right. What they then did was then they put it into a centrifuge and they spun it down, and they took a look at where these new strands of DNA would position itself. Would it fall all the way to the bottom? Now, if all of them always fall all the way to the bottom, that means all of the new strands of DNA would be much, much heavier. But if all of these new strands of DNA was always staying on the top, meaning that they are much lighter, like in the case of here that you see in this band, well, that means they're incorporating the N14 instead. But if they use if they actually incorporate just parts of it, meaning they keep some of the old strand and then they have some of the new ones combined together, then they would have an intermediate weight, which is what you see over here, indicated in purple. All right. Now, here's the thing. When they did this experiment, what they found is that if you propose a conservative model, so you are a believer that the DNA will replicate using the conservative approach, meaning that you would have the original strand that would only keep a copy of itself, and then the new one would always be incorporating only new nucleotides, meaning based on all of the new material that you provide with it, right? Then what you will see would be two distinct bands. One band based on the original DNA molecular weight, and then the other one based on the new material that it has been built from, right? And this is what the end result you would expect to see. But then what they found was that this did not happen. Instead, after subsequent generations, what they found was the new DNA material that were produced were more on the intermediate density level. And as time went on, meaning more generations were allowed to continue to grow and reproduce itself, you will find more and more a higher concentration of bands that were within this particular range, meaning on the intermediate and then the lighter range, because because of the material that you're giving it. Whereas if you were a believer in the dispersive model, then what would happen is that it should only have one type of density. Why? Because remember, here in this schematic diagram, you would be expecting sections of the new DNA combined with sections of the old DNA. So essentially, they would all come out to be the same density. You wouldn't expect a variation in density. So that was how they used it to figure out that it was the semi-conservative model that is the way in which DNA replicates itself and not the other two. A lot of what I have just described, it's just a simple overview. Go ahead and watch the video that I had just given you, which will really help you, because the visual aid in that video, I think would be most helpful for you to follow through how you can do it. So this is the video that I am talking about, and I have given you a series of questions to answer as you're watching it. You may find that you need to watch it 1 or 2 more times in order to understand the scientific data that is proposed. This particular slide, although I couldn't remove the source of it, but which is fine, all right? Hopefully this would also help you visualize what I had just tried to describe using the 2 slides before that I tried to explain here, all right? Where how the scientists actually figured out that the conservative model and the dispersive model, neither of these two was the approach that the cell actually would use for copying itself. It actually is this third one, the semi-conservative model, because of the types of bands that they would see. Now, I wanted to show you one more thing, and that is this. If you are planning to take the SAT2 exam, very often they don't always give you diagrams. And if they do, it would be more likely that they give you data table with numbers or graphs. So this is the experimental results, portions of it that was recorded using based on the Meselson-Stahl experiment. After when you're done watching the video, I would like you to take a look at this to see if you can understand this. Now remember, when you take the SAT2 exam, they give you 60 minutes to answer 80 questions. So you have to be able to kind of almost know what you're looking at the moment you see this question, and not spend too much time analyzing what's really happening in here, all right? And again, this visual aid, I have found it quite helpful, and perhaps this is something that you and I can go over as well in the next review session that we have on Thursday. All right, I have also found this particular shortened version of the Meselson-Stahl experiment. If you have, go ahead and try the other one first, which is where the Q&A is all based on, and then see if you can, if this would help you. What I found is that when I looked at and compared many different video clips, they each have their own advantages and disadvantages in terms of student comprehension. I certainly like the 8-minute one that I gave you more, because I thought it was a bit more, it was presented a little bit more clearly compared to the other one, but this one is a decent overview too. Maybe you should watch the other one, and then when you have time, go take a look at this 2.5-minute one. All right, so let's talk about how this actually happens on a molecular scale, all right, on the cellular level. Now, here is the schematic diagram of a double helix of the DNA. Essentially, what has to happen during the process of copying DNA, one portion of this DNA would have to unwind. The double helix have to unwind, and this is all handled with the help of many different kinds of enzymes. You unwind, and there are enzymes that would hold the unwind position in place, all right, allowing other enzymes to come in and add the free-floating nucleotides that are already inside our cells, and you slowly add the base pairs that should cross-match with each one. Now, you should notice the color coding. There's a pattern to this, right? Red color binds with the white, the light yellow binds with the orange one. The whole idea is A pairs with T and C pairs with G, because that's how the base pairs would actually form. That was what we observed. So there are enzymes that are helping the DNA copy both sides of the template that are exposed. The templates are the gray lines that you see here. Those are the original DNA. And then once one section of the DNA that has been unwound is fully copied, then the enzyme moves down towards the next section of the DNA, unwinds another section, and then holds it in place, while these other enzymes that are here will continue to add the new nucleotide bases and then combine them together with the help of another enzyme until you go all the way to the end, right? So imagine for a moment that you have a zipper, that you and I can physically hold, but then somehow this zipper is twisted into a helix form, and that's the natural setting that it has. But in order for you to unwind it and unzip it, you've got to have something to hold it and clip it in place so that it won't want to snap back to its original position, which is the helical position, right? The twisted position. So this will continue on all the way until the end, until you have two copies of identical DNA. Now, you should notice right away that on one strand, you have the original gray color strand, which is reminiscent of this original DNA chromosome that we're trying to copy. And the other half of it is made up of the new strand, which is indicated by the light blue and the dark blue strands that you see here. But both copies are identical. And this is what the cell cycle allows a cell to do. So let's take a closer look at what really happens in terms of the cells. So here are three basic types of enzymes that are involved here. When you watch the video, there will be more enzymes that will be introduced to you, and I would also slowly uncover the rest of them for you too. But let me help you out with a little bit of the vocabulary here. DNA helicase is actually the enzyme that I mentioned at the very beginning that separates and unwinds the DNA double helix, all right, to form the replication bubble, which I'll show you in the next slide. DNA polymerase is, now there are many different kinds of DNA polymerases. This is just a very general term that is being used here. The DNA polymerase synthesizes, meaning it adds the new nucleotides to the original old DNA so that you can form the new strand, all right? And then you have DNA ligase, which acts like a glue that combines all of the individual sections of nucleotides that has been newly added to the old template. They form what is called, each of these new sections are called Okazaki fragments, and you then glue them together so that you have a continuous strand of DNA. This is a lot clearer when we look at it in picture form. Let's take a look at this. So here it is. Imagine here is a double helix, and I am interested in copying the DNA material that is here. What I would do is that where the DNA itself unwinds, it forms what is known as a replication bubble. So because you've unwind it, you are now expanding it, and you're breaking the chemical bonds that normally holds the DNA nitrogen bases on both sides together. But right now the bonds has you have opened it, and then what it does is that it creates what is known as a replication fork. Now, if you could imagine, if this was a rope that was all twisted and you have to physically unwind one section of it, right? And then you kind of slide the two sides of the rope open because they are tightly coiled together, you should be able to imagine that right away on each of the downstream or the upstream of this DNA double helix, it would have a lot of tension, and that it would eventually you would have to unwind and reduce the tension behind those as well. So now, if you take a look at this, the DNA replication process, right here, this is the fork, the area that has been opened. If you take a careful look at this, right, the DNA double helix is anti-parallel, meaning one side moves up in one direction, the other one moves in opposite direction. Remember at the very beginning of the lecture, I showed you an image of the ACTG where the author had written all of the letters so that it's easy for you to read. However, in reality, I told you these bases, if you were to manipulate them as a molecule or on a piece of paper, you would actually have to turn it upside down. Well, this is what we're looking at. So this particular strand of DNA, the 5' is here, that starts on one end. And then if you follow my cursor, it then goes on to the other end, which is the 3' right here. And then on the opposite side is anti-parallel. So 5' would be here. And then if you follow this cursor, then the 3' will be on the other hand. So when the DNA is being copied, one side of the strand, the side that has the 3' that is exposed right here, will be what we call a leading strand. Notice how the arrow is a continuous arrow, meaning that you can keep copying DNA all the way towards the fork, towards the inside of the replication fork without stopping. However, on the opposite side down here, you see where this 5' in is right here that you're trying to copy? These arrows are not continuous. They seem to be start, go, and stop. Start, go, and stop. All right? There's a lot of stopping and going. Why do you think that's happening? Why are the DNA polymerases, which are the enzymes that are responsible for helping you make copies of these new DNA strands, why do they work in opposite directions? How come this bottom strand doesn't keep copying this side, especially if you imagine this is the side that you're trying to unwind and unzip, right? Why do you have to go stop and go back over and over again? Well, because there's something interesting about this that you must take into consideration. All right. The DNA polymerase that is copying the DNA, it builds a new DNA strand always from 5' to 3', which means where it begins to copy this strand, the new strand that is building, that's going to be the 5', and it always keeps building towards the 3'. As long as it can do that, it will be a continuous strand. However, what that means is that where it is trying to make a copy of, it is moving along the old strand, which is represented by this light or brighter blue color. It's going to be going from 3' to 5'. In other words, you are copying it. The strand that you are making would be the 5' that you're starting. And then as you keep adding new nucleotides, meaning the A, C, T, and G that can base pair with the whatever is being exposed on the blue line right here, that would be moving towards 3', which is moving towards the inside of the fork. However, if you look on the bottom strand, which we call the lagging strand, you have an anti-parallel scenario going on. This side is actually the 5', not 3' like this. So in order for the DNA polymerase to move along the unzipped portion of DNA, all right, so that it can be going from 3' to 5'. Remember, the 3' is somewhere over here at the end, right? And you trace this again. So 3' is somewhere over here, and you're moving toward 5'. So you're using this old template, which you have to move by reading it from 3' to 5', such that when you build a new strand, indicated by the darker blue color here, then you will be moving the right direction of 5' towards 3'. Now, it is discontinuous. Essentially, the DNA polymerase, there will be a primer, essentially an enzyme, a molecule that would be standing by right here, letting the enzyme DNA polymerase know, hey, this is where you're going to start copying this portion of the DNA. And then it can only travel in this direction, meaning the DNA polymerase can only travel in that direction. So then it will be copying, building DNA from 5', it's on the new strand, going towards this direction, because then this side would be its own 3'. But once it get down here, it would have to come to a stop. And then the primer would unlatch itself, add another one, put it right here, so that the DNA polymerase will then move back here and say, okay, I didn't copy this side yet. I will move down here and then copy down from 5' to 3', and then will unlatch and then come back here and then add another one down here. Now, you would notice that there are gaps in here. Each one of these gaps where the primer used to be, new nucleotides will have to be added back in, and then all of the gaps will then have to be chemically bonded together with the help of DNA ligase, which is that third enzyme that I mentioned, all right? And now, some of you may be thinking, but why is it so broken the way it's being copied? Remember, this DNA double helix used to be coiled around itself, much like the way you see this section. You're doing it in small bits and pieces. So you don't unwind a large section of DNA, you uncoil a little bit at a time. So the moment when you open this area, right? Imagine if the back of all this, where this is now open, imagine for a moment that it was still twisted, right? And you only open this section. Well, the DNA polymerase will then recognize, with the help of the primer, that this is the 5' end, where the new strand would be, and it would start copying toward this side. Why? Because all of this section is not unwinded yet. Until this section is unwind, then the primer will then come here, attach itself on here to signal the DNA polymerase, hey, this is where you should be copying. This is the 5' end. Well, your new 5' end. And then we'll start copying all the way down here. And then when the next section opens up and unwinds, then the primer will be established right here, allowing the DNA polymerase to move back towards the inside of the replication fork, and then going towards 5' to 3' again. All right. Now, that having said, on the top, because the DNA strand on the top, the DNA polymerase, it's always moving from 5' to 3'. It can keep copying as long as the DNA unwinds itself. So as you open up the two sides of the DNA, the copying of the DNA can be continuous, keep moving in one section at a time without stopping, which is why this side are not going to form what we call Okazaki fragments, only on the lagging strand side, which you have these sections of DNA that are newly formed, that still needs to be combined together with the help of DNA polymerase, which is why this is called the lagging strand. And that one on the top is called the leading strand, that is continuous. Okay, here is another way of looking at this from a different kind of drawing that I thought for some of you may have an easier time, where you can see that there is the primer, here's the DNA polymerase. Of course, the one on top is the leading strand, and this DNA polymerase is going from 5', meaning the new strand that's being formed, going towards the 3'. Now, some of you may be thinking, well, how can you tell this is this is pre-labeled for you, but on an exam, the 3' and 5' oftentimes would also be labeled, so you can figure out right away which side is the leading or lagging strand. But then on this section, right, you notice how the primer latches onto here, allowing the DNA polymerase to know that, hey, we have to start copying right here, so 5' will be here, moving towards this end to the 3' end, until this particular enzyme here unwinds the next section of DNA. And of course, the DNA polymerase number 1 on the top will just keep copying, no problem. It's already very smooth sailing. But then for this one, this primer will have to move back one more step in order for it to know that, hey, I have to move back here and then copying and finish this. That's actually how it is done. All right. And by the way, so there are a bunch of enzymes that are not mentioned in here, like topoisomerase, which is going to be mentioned in the video, the primers, and the different kinds of DNA polymerase 2 and DNA polymerase 3. Okay, so then after, when it's all done, the DNA ligase comes in, and once the new nucleus has put together, it will help form the phosphodiester bonds that forms where the two sides of the DNA is, and the hydrogen bonds will be formed between the nitrogen base pairs that you see right here. All right. And this is a simple schematic diagram that you have seen before. This is just a quick reminder of what's really going on. All right, the semi-conservative model. I have taken a look at the Amoeba Sisters illustration. It's a bit simplified, which is why I have chosen to use this. But now, go ahead and use this to answer the questions. If you want to, you can certainly go ahead and use Bozeman Science or crashcourse.com to help you answer some of the other questions if you find it to be easier, and that is fine too. Okay, and in the next portion of the lecture, what we will do is we will engage and talk about mutations, various types of mutations, how it occurs, and why it is dangerous. But in this particular last slide, what I just want to mention to you is that as the DNA polymerase copies DNA, they actually have proofreading mechanisms where they will make sure that the copying of the nitrogen bases are correct. And whenever they find something that is wrong, they would actually have an enzyme to cut out that portion of the nucleotide, which is miscopied, and replace it with the proper one to reduce the chances of genetic mutation, which is really amazing. And the number of mistakes happens at about once per 1 billion base pairs, which is superb. All right, I'm going to get ready for your next lecture later on today. So hopefully you will find the video clips helpful, and I look forward to talking to all of you when we do our review session on Thursday. Take care, everyone, and stay safe.